Hello, my name is Hugh Laverty, and I'm the Head of Scientific Operations at the Innovative Medicines Initiative. Before I start the short presentation today, I'd just like to remind you that the views presented here are my own personal views, do not reflect the position or policies or any of the organizations that are mentioned in this presentation. As I said, I'm the Head of Scientific Operations at the Innovative Medicines Initiative. And we are a public-private partnership between the pharmaceutical industry and the European Union, and we fund projects that are focused on tackling key bottlenecks within the drug discovery and drug development process. The important thing about our projects are that these are multidisciplinary collaborative projects between public and private partners. We were set up originally in 2008, and we were renewed in 2014. And across these two programs, we have had a budget of about €5 billion and funded to date about 150 projects. We have a match funding model, so 50% of our funding comes from the EU and 50% comes from industry. What's very important about our model is that none of the public funding actually goes to the pharmaceutical companies that participate in our projects. They actually have to bring their own resource into those projects, a thing that we called in-kind contribution. As I said, our projects are working on trying to develop new tools, new methodologies that will speed up and overcome these bottlenecks within the drug development process. Our projects are not working on the development of new treatments per se. We have a few projects in the area of antimicrobial resistance and also in infectious disease in areas such as Ebola, where we are working on compounds. But generally, we're working on tools and methodologies. And we hope that these tools and methodologies are going to feed in to the drug development process, and that this will then accelerate the process by which patients get access to new treatments. Now, I've been asked to say a few words about how we take innovations in the pharmaceutical sector from the bench through to the market. And I focused simply on how we might take a new medicine, because I'm using this as an example because time simply does not allow me to go into all the different variations that are possible. And this slide summarizes the main steps, but it is at a very high level. And it's a very high level view of what is a very complex, very long, and ultimately very expensive process. Now, normally when you start medicines development or new treatment, it is based upon a lot of early discovery work. Researchers, clinicians have worked for many years on understanding a disease, and they have identified a molecule or a treatment that they think is a good candidate for drug development. At that stage, the candidate molecule will then move into a phase of preclinical studies, which is a much more structured way in which that we try to understand the behavior of the molecule in mammalian systems, either at the cellular level or within animals. And we're asking questions such as, where does the molecule go in the body? How is it absorbed? How is it metabolized? How is it excreted? What are the metabolites? How do you deliver it? How do you make it available in the right tissue at the right time? And very importantly, before you can go anywhere near humans with your molecule, you have to do a series of studies in animals that show the toxicity of your molecule is safe, or basically there is no toxicity. And this is a set of acute and chronic dosing studies that really tests and determines whether there are any safety signals associated with your molecule. You then take this package of data, you go to the relevant national competent authority, usually a regulatory agency, and you say, can I do my studies now in humans? If you get approval to do human studies, you go sequentially through phase 1, phase 2, phase 3. Phase 1 studies are very much focused on testing the safety of the molecule, and these are done in healthy volunteers. If everything goes well and there are no safety signals, you may then get permission to move to phase 2. And at phase 2, you're beginning to test the efficacy of your molecule, but in a patient population, so patients that have the disease. You're also trying to determine what is the best dose to use your molecule at. Again, if everything goes well and you have no safety signals and you have good efficacy, you may get permission to move to phase 3. And in a phase 3 study, you're actually taking the molecule at the most appropriate dose and exposing it to a much bigger and a much wider population. Again, the focus here is really on efficacy, but safety is always been checked and monitored. Now, based upon the outcome of your clinical studies, you may have a portfolio of data from your preclinical and clinical development. This will also be complemented by data that you have generated regarding the manufacturing of the compound, because you have to show that you can manufacture the compound at scale, it's a reproducible process, and you have robust bioanalytical methods. So you will take all of that data and then you will submit it to the regulatory agency and ask them for an opinion, ask them whether you can get marketing authorization or not. The regulators will use independent panels of reviewers to review your dossier, and they will determine whether you can get approval so that you can market your compound, your drug, on the market, or they may determine that there are weaknesses within your dossier and therefore you have to go back and you have to do more studies preclinically, clinically, of a specific type, and then you will have to resubmit that dossier. Even when you get a molecule to market, the molecule is still, and the use of the molecule is still monitored in a process called pharmacovigilance. And so therefore, if any unexpected safety signals or adverse events are associated with your molecule, this information is fed back to the regulators, and they will make an assessment on the benefit of keeping the molecule on the market and the risk to patients from the safety signals that have been observed. In extreme cases where you get serious adverse events, the regulators often decide to withdraw the molecule from the market and it is no longer available. At all stages of this process, safety is the key concern in both preclinical and clinical testing. And the regulators are always making this assessment of whether it is beneficial for a drug to reach the market or not. And so at every stage of the process, you need to seek independent review of your data and authorization from the competent authority or the local ethics committee or the regulatory agencies. And as I've said, even medicines that are on the market are still subjected to pharmacovigilance in case unexpected signals may come up. And as I've said before, at all stages of the drug development process, safety is of paramount importance, and the regulators are always ensuring that the benefits of having a medicine on the market outweigh any risks that might be associated with it. Now, I have, what I've described today has been quite a high-level view of a very simple drug development process. Now, there are other processes involved, depending on the indication, whether this be in orphan indications or dependent on the patient population, such as within pediatrics. So there are variations and there are differences in the way in which you might seek regulatory approval. However, but for more information and for more detail on this, you should go to the website of your national regulatory agency. Every country has a national regulator, and they will provide you more information. I have just shown 4 of the well-known agencies that I am aware of and I have been in contact with in the past, But as I've said, every country has a national regulatory agency, and you can find a lot more information about the process, because I have just given you a very high level view. So thank you for your attention, and I look forward to the panel discussion on the 30th of April.